So again, I will start by saying what was briefly said moments ago. So my name is Katherine Czysz, and I'm a senior field application scientist at Fujifilm Cell Dynamics, or FCDI for short. And I've been working with stem cells and stem cell differentiations for over 14 years right now. And the last 6 years I've spent with Fujifilm Cell Dynamics, again, FCDI. And that gave me an opportunity, because I work with customers globally, that gave me the opportunity to see how our commercially available human iPS-derived cells are adapted globally in all research areas that you may think of with regards to in vitro and in vivo settings. So today I have a really great opportunity, thanks to the BRIC community, to talk to you today about our commercially available human iPS-derived cells that you can use for efficiently generating, again, human-relevant 3D models for drug discovery and disease modeling. So we're gonna see lots of data today, and I welcome all the questions moving forward. Okay, so obviously, as I mentioned, we are Fujifilm Cell Dynamics. So Fujifilm is the company that all of us know for cameras and, again, their global presence. So I will just give you a little bit of an overview of how Fujifilm Cell Dynamics is fitting within Fujifilm company. And again, you can see that with Fujifilm corporate summary slide, you can see again that we have a global presence worldwide. And this kind of like infrastructure allowed us to, again, have a global view with expanding number of life science companies that Fujifilm is acquiring. We have a global view on again, how our cells are being used in all research areas. And also with Fujifilm life science companies portfolio listed here, we provide a lot of different research areas coming together via, again, our Fujifilm Cell Dynamics commercially available iPS derived cells, but also with Vaco chemicals that all of us have probably used in our lab-based research. With Vaco, I'm also very happy to say that Vaco is again, a great company that all of us used for chemicals reagents, but also Vaco is kindly distributing our iCell products in Japan as well. They're also in great contact with BMS, who is actually your distributor in Korea as well. So this is something I will mention later on as well. But we also have a number of additional companies, like massive companies like Irvine Scientific and Diosynth, but also Shenandoah, our newest addition. So all together, with regards to what our companies provide, we come together to provide you lots of solutions to your daily lab research. And with regards to Fujifilm Cell Dynamics, we've been acquired by Fujifilm in 2015, but as a company, Cellular Dynamics, we have been generating iPS-derived cells since 2008, 2010. So we have a lot of experience, again, with regards to bringing you human iPS-derived products that has been, again, as I mentioned before, globally adapted in safety toxicology, drug discovery, disease modeling, cell therapy, and so on and so on and so on. As a company, we do get to enjoy 2 headquarters, because obviously Fujifilm headquarters are in Tokyo, Japan, but our Fujifilm Cell Dynamics headquarters are located in Madison, Wisconsin, in the United States, where our production team generates cells which are then cryopreserved and shipped globally. And again, BMS is pretty much your perfect distributor to efficiently provide you with the cells in Korea. So again, I'm happy for you to be able to connect with BMS and ask all the questions later on as well. So with regards to what we actually have available with regards to our commercially available human iPS-derived cells, we usually categorize our products. We usually categorize our products into 3 major groups. So on this slide here, I'm showing you, again, those 3 groups being divided into a general group of iPS-derived cardiomonocytes, iPS-derived neural cells, so neuronal and glial cells, and also hematopoietic and immune cells like hepatocytes, macrophages, and so on. So again, all of these cells have been widely adapted globally via, again, all the research areas And what I like to highlight here is the fact that we do provide a lot of application protocols, a lot of user guides that support pretty much any assay you may choose to use these cells for. And as a company, we are very, very proud of all the technical support we provide. So again, with BMS and their support, you also can easily access technical support and all the documents for your assays of interest. And I'm not sure how many of you have worked in 3D models from the practical standpoint. But again, as you can anticipate, the 2D model is obviously the monoculture in 2D has been widely adapted in drug screening, disease modeling, because it's really fast, efficient, and very applicable to screening technologies. But over the last couple of years, again, with customer-facing interactions globally, we can see that it is more and more possible, and it has become easy to establish screening platforms in 3D. And this very general overview slide is showing you that, again, monoculture is rapidly expanding into, again, co-culture systems in 2D, in more advanced culture settings like transwells, but also scaffolds, obviously organoids, spheroids, and obviously extrapolated out to the in vivo situations as well. And this simple snapshot here shows you just pretty much the search results from seeing how many publications are focusing on 3D spheroids and organoids over the last years. Again, from the customer-facing point of view, I can tell you that over the last 2-3 years, we've seen explosion of interest in doing screenings and disease modeling in 3D systems. And our application team, our R&D teams have supported that beautifully. And we now have a wide range of applications that allow you to very quickly build 3D models, build 3D models in high throughput screening, high content analysis friendly, functional based setup. So before I start showing you some data, I also wanted, for those of you who are still unfamiliar with what our portfolio offers, I wanted to show you this very, very general slide showing that, again, with regards to the brain and nervous system, we have a number of different wild type and disease models focusing on glial cells, on like astrocytes and macroglia, but also blood-brain barrier, and a wide range array of neuronal cell types like GABAergic neurons, dopaminergic neurons, glutamatergic neurons, motor neurons, our newest addition of induced excitatory neurons, which are very, very cost-efficient solution for screening, and more to come, and actually sensory neurons as well. This is something I will mention in a moment as well. I will also show you some beautiful data on our cardiac portfolio, how easily you can mix cells like cardiomyocytes, endothelial cells, our cardiac fibroblasts, all of them coming from the same donor of iPS cell donor. And you can use those spheroids for 3D screening. Again, the same with hepatocytes and, for example, with macrophages for those of you who are working in hepatoimmune field. And this is just one of the last slides from the introduction part. I wanted you to have access to this slide that shows some newest addition to our expanding portfolio. As I mentioned moments ago, the sensory neurons is our newest addition to your portfolio. For those of you who are working in pain, I encourage you to contact BMS. I will share the contact information with you later on, and inquire about availability of these cells. They are beautifully described, they are beautifully functional, and again, it's a pleasure to be representing these cells and seeing how well they are becoming adapted by our customers globally. And again, you can see a lot of additional information with regards to additional cell types that we have recently released in our portfolio, and also some cells which are actually becoming available now. And I can see that actually I need to update this slide because induced excitatory neurons and astrocytes are available off the shelf right now. And again, induced excitatory neurons, for those of you who again, who are looking at excitatory cells for 3D, so 2D screening applications, please again let us know, because these cells offer a really extra actually efficiency per well with regards to screening. So again, great solutions to many different setups. So as you can see from this slide, throughout this presentation, I will show you some of our really nice data, typical data for our ISO neurospheres. So we will start by discussing how you can combine specific cell types to create pretty much mini brain spheroids, and how you can use them for screening and for disease modeling. I will also show you some beautiful data on our ISO cardiospheres, where again, you can mix cardiomocytes, endothelial cells, cardiac fibroblasts, all of them from the same donor. And you can quickly set up this screening platform in your lab. I'm going to also show you some initial data on how to generate BBB spheroids from our BBB model, consisting of BMEX, obviously, astrocytes and pericytes. And for those of you who are working on hepatoimmuno field, or even immuno with macrophages only, I will show you that it is really easy to generate those spheroids, again, for the high throughput screening friendly manner. So let's start with ISO neurospheres. And again, this slide just shows you that, again, historically, most of the research have been done in 2D in monoculture, right? Because we are used to having one specific cell type, 2D screening, and job done. But now, over the last couple of years, especially again with better kind of like cohesion of great cell types that we provide, and also a great screening platforms that are available there that we have a lot of information on, we can easily now extrapolate this monoculture in 2D and co-culture and three-culture systems in 2D, and also extrapolate this 2D to 2.5D, 3D, organ-on-chip, microfluidic systems, and so on. So today I will focus on 3D systems, but whatever your application is, if you're working on microfluidic systems, organ-on-chip, always let us know, because we have thousands of publications on our cells. We are extremely established globally, and our application team is constantly developing new protocols and new updates and so on. So lots of support from our side, regardless of assay of your choosing, for high throughput, mid throughput, and research areas. So this slide pretty much shows you that generating those ISO neurospheres is incredibly, almost ridiculously easy. Again, before joining CDI, I worked a lot in 3D systems, and I suffered a little bit. And I was delighted to see that with regards to generation of ISO neurospheres, not only you have the ease of setup, because you pretty much on the day of thawing the cells, because our cells come to you cryopreserved, so you pretty much have them in liquid nitrogen, you store the media separately, and then when you want to thaw the cell, when you want to start working with the cells, you simply thaw the cells, follow our protocols, and there you go. A couple of days later, a couple of weeks later, depending on the assay, you can start using the cells. But for the generation of ISO neurospheres, you can just pretty much choose the cell types of that you're going to mix together, and you can pretty much thaw them together, set up the spheres. And 2-3 days later, you can see the whole plate set up, beautiful reproducibility, well-to-well. And 2 weeks later, you can start adding your compounds for, again, high throughput screening, disease modeling, and now you will see some beautiful data. So you can pretty much mix and match any cell types that you are interested in, and I'll elaborate on that later on as well. And again, generating those neurospheres provides you with a lot of control with regards to the cell makeup. You control the cell ratios, so, and we have lots of information on kind of like direction, depending on the direction you want to go. So we have lots of expertise and lots of advice for you how to move forward. You can easily create specific disease models that allow you to compare the wild type and these disease models in 3D side by side. And using functional and ICC assessment, you can compare how this wild type and disease model differ, and you can screen the systems for your against your compounds and see which compounds are reversing the disease phenotype back to the wild type ones. So again, beautiful setups, incredibly reproducible system, and we are now working on on adding macroglia to neurospheres, which are usually consisting of neurons and astrocytes, etc. So again, IsoNeurospheres is a uniquely accessible and modular method for generating advanced 3D brains models, because again, you control by the cell makeup the regional part of the brain you want to focus on. And again, it's super simple assay. I'm still amazed by how easy it is to have this adapted. And we have a number of beautiful success stories from around the world showing us, from our customers, showing us that you can easily build this platform. You can easily use it for screening, and the data looks beautiful as well. So you will see from the next slide, I think, that again, we have a number of different cell types that allow you to focus on specific disease models. And by doing so, you can compare, again, the healthy versus wild type cells very easily. And it's just pretty much as easy as mixing different ratios of glutaneurons, dopaneurons, astrocytes, GABA neurons, again, whichever the direction is that you go into. And you have options for, again, substituting specific cell types from wild type cells, healthy cells, to the disease models. And assay systems are super easy as well. You have an array of assays you can choose to focus on, including functional assessments of cells by calcium imaging. So again, calcium fluctuations analysis, and also electrical activity of the cells on MEA, and also obviously ICC, so immunocytochemistry for high content readout. And this is a very simple slide showing you how beautiful a 384 well plate system can look like, with only one spheroid per well, incredibly consistent across all the wells and rows. And you can see that you can pretty much even use A1 well versus pretty much P24 and so on. All the edge wells can be used as well. And again, it's incredibly reproducible platform. In this specific setup, the team has mixed glutaneurons, GABA neurons, and astrocytes, and pretty much one vial of glutaneurons, one vial of GABA neurons, one vial of astrocytes have been easily mixed within minutes together after thaw, and pretty much the cells were added to wells accordingly. And 2 days later, you can see beautiful spheroids that again are high throughput screening amendable 2 weeks post setup. And reproducibility is everything to us, because it's really important to be able to set up experiments in different timelines at your convenience. And reproducibility is something that we always pay the closest attention to. And this is pretty much a very simple slide showing you that you can, by following our protocols and lots of guidance that we have, you can actually control the diameter of those spheroids really tightly. There's no necrotic core or nothing like that. And you can see that if you, for example, mix 22,500 of glutaneurons and GABA neurons all together, you will have a very specific range of like diameter of those spheroids. If you add additional 2,500 of astrocytes, as shown here, again, the diameter goes up a little bit, but again, look at how reproducible the information is. And this is really consistent across the plates, across the setups and different times and so on. And we, again, help you a lot. And our protocols are very simple, very easy with regards to the controlling of that size. And we provide a lot of reference information as well. And I keep mentioning that this is easy. It is easy to set up this assay, and it really is. Again, you order the cells. It's super easy for you to get these cells from BMS, again, our local distributor. And then once you have the cells in the lab and you're ready to thaw them, it's pretty much very simple, very short setup on the day. You mix the cells together. And then 2 weeks later, not even 3 weeks, 2 weeks later, you can start using the cells for assessment. Again, this differs drastically from, again, do-it-yourself brain organoids, do-it-yourself neurospheroids, again, and the cells are highly functional, highly reproducible, and again, just beautiful to work with. And this is pretty much extrapolating of the bird's view on the morphology of the cells in the plate upon adding calcium dye to the cells, to the spheroids. So again, it's a simple addition. 2 weeks after spheroid setup, you add the simple dye, the calcium-6 dye, and you can see that all the spheroids are lighting up and very reproducibly. And then you can use platforms like FDSS from Hamamatsu, Flipper from Molecular Devices, or any other calcium imaging platform that will allow you to measure calcium oscillation over time. So this is one of the super high throughput friendly platforms that again allows you to look at calcium oscillations patterns which in turn allows you to have this functional readout and compare again wild type and the disease models side by side. There are other functional readouts like MEA. There are other high content readouts like ICC that you can choose here as well. And we provide lots of support on that. So it's super easy adaptation. So this is finally first slide with data. So thank you for your patience. And you can see that this slide is divided in two, right? So you have left side with the data from Fujifilm Cell Dynamics. So our site generated this data on our isoneurospheres. And this data, row by row, compares the data from the NCATS publication. So NCATS is pretty much an organization on the East Coast and States. Really, really cool data produced from this group that were published in this strong publication from 2022. Shows that you can simply, again, mix and match our cells at different ratios to represent different regions in the brain. And by using, again, calcium oscillation platforms for functional readout, you can pretty much modulate the calcium oscillation patterns. And those patterns are very producible depending on cell types and the number of cell types that you mix together. So you can, for example, mix cells and similar in the ratio shown here to have like VTA-like brain regions to focus on Parkinson disease modeling. You can have a prefrontal cortex, right, set up to focus on Alzheimer's disease modeling and so on. And again, we do support a lot of that research moving forward. So for now, we're gonna, for a moment, focus on disease modeling with isoneurospheres. Again, high throughput screening friendly manner. And again, as I mentioned, you can very easily build neurospheres from the wild type cells and the disease models. And this is very easy with the portfolio of the cells we have. This is just a very short representation of the extensive number of products we have for disease modeling with our neuro portfolio. And you can see here, so for example, for Parkinson's disease, you can compare healthy dopaneurons with Parkinson's disease dopaneurons, and again, have them side by side compared for calcium oscillation differences or ICC or MEA differences. The same for Alzheimer's disease, the same for ALS and FTD, and the same for neurodevelopmental disorders, including actually epilepsy modeling with our KCNT1 and SCN1A models as well. So a lot of data, not only from us, but also from, for example, molecular devices on the Flipper platform, Hamamatsu, and so on, and most importantly, from our customers who are very happily adapting this platform globally. So this is a beautiful, beautiful, beautiful slide from this strong publication from NCATS I showed you earlier on. And just look, hopefully you can see the cursor, the mouse I'm using to hover over the slide. And you can see here on the top left part of the slide, highlighted with a green line, you can see calcium oscillation patterns of isoneurospheres, which were developed by mixing healthy dopaneurons and astrocytes. And you can see here that upon treating these neurospheres with DMSO, the cells again are having really nice, healthy pattern of calcium oscillations. But if you substitute those healthy dopaneurons with our Parkinson dopaneurons, with a simple point mutation, A53T point mutation in alpha-synuclein gene, so the mutation linked to PD development, you will see that those neurospheres with this Parkinson disease model of dopaneurons having severely disrupted calcium oscillation patterns upon just pretty much vehicle treatment, DMSO treatment. So you can see that the healthy cells have a beautiful pattern. DMSO treated Parkinson disease models have very disrupted pattern as shown here on the slide. But interesting thing is that when you add ropinirole compound, which is pretty much clinically approved anti-Parkinson disease treatment that is pretty much focusing on dopamine agonist receptors, you can see that even at low concentrations, this ropinirole compound is actually reversing the disease phenotype back to the normal one. So again, like upon treatment with ropinirole, which has been associated with impacting calcium oscillation pattern, making them healthy, you can see that upon this positive control compound, the baseline is coming back to represent the wild type as well. So this is a beautiful observation, and again, It's highly reproducible. We have lots of data on that. And the same goes for modeling Alzheimer's disease. The same principle applies. You pretty much generate isoneurospheres, having our isogaba neurons, the healthy one. So all of our healthy cells have an ApoE33 isoform. So the typical normal healthy ApoE33 isoform. And you can see that those healthy isogaba neurons with ApoE33 isoform have a beautiful normal calcium oscillation pattern. This is the data that in red, you can see the data derived from our team. And again, you can see a beautiful calcium oscillation pattern for ApoE33. But if you substitute these healthy GABA neurons with ApoE33 isoform for isogaba neurons with ApoE44 isoform, which all of us know is highly associated to Alzheimer's disease development, you can see that this basic oscillation pattern of calcium oscillation is drastically disrupted, because again, ApoE44 has been shown to actually impact calcium oscillation patterns in the cells, because ApoE44 has been actually shown to impact NMDA receptors and impact glutamate homeostasis in the cells, which is actually associated with Alzheimer's disease development. But you can see that when these ApoE44 isoneurospheres are treated with memantine, the clinically approved anti-Alzheimer's disease treatment that actually targets NMDA receptors and blocks them, therefore maintaining glutamate levels and maintaining calcium oscillation patterns, you can see that this pattern becomes more and more similar to the wild-type isoform. So here you have a beautiful pattern for healthy isogaba neurons with ApoE33 isoform. You have disrupted pattern with ApoE44, because again, ApoE44 is highly disruptive to calcium oscillations. And when you treat this disease model with memantine, you can see that this anti-Alzheimer's disease compound treatment is actually reversing back the calcium oscillation pattern, is repairing calcium oscillation pattern back to the healthy one. And this is in red, our data from Fujifilm Cell Dynamics. And you can see below the data from the publication I mentioned before. And again, it's highly reproducible system. So again, side-by-side comparison across different sites. We have plenty more of data from our collaborators, customers, showing exactly the same pattern and exactly the same reproducibility. So it's beautiful. And again, it's just a snapshot of what you can do. So always let us know, let BMS know what your interests are, and we can help you have that adapted in your lab in no time. The same for epilepsy. If you are studying epilepsy, again, it's very easy to compare the healthy versus epileptic models, have some compounds thrown on the cells, do the screenings, and again, high-throughput screening-friendly manner. And this is the data from molecular devices. They have also lots of data on Alzheimer's disease modeling as well, but I'm more than happy to talk about it offline. And again, highly reproducible pattern of setups. So now we're going to switch the gears, and I will quickly tell you a little bit about our blood-brain barrier for those of you who are working on analyzing which compounds can actually penetrate blood-brain barrier. If you're looking at recovery, penetration, or pretty much disease modeling as well, this is pretty much the system for you. It's incredible how well adapted this ICEL-BBB model has been since we launched it globally. And again, beautiful reproducibility. And for those of you who are actually working on BBB models or who are interested in BBB models, please ask us for more information, because we have, again, lots of data on this. And in general, we have adapted a number of different assays for these cells, starting obviously from the historically approved transfer system, where you have like the cells, BMX cells, the main cells of the BBB system, and site of the transfer, and our astrocytes and pericytes actually immediately below, to be honest. So again, historically, we have lots of data on transfers, but also we have a lot of interest on microfluidic devices, and we're starting to develop more and more data on 3D spheroids as well. For those of you who are interested in this model, do let us know. And our BBB model compares incredibly with regards to their superior functionality, in vivo-like functionality, when you compare these cells with any other available model on the market. So again, if you have a BBB interest, let us know, because this is incredible groundbreaking model that will provide you with batch-to-batch reproducibility and highly functional data as well. So the system contains of the 3 cell types, so BMX, so brain microvascular endothelial cells, obviously the main cell type of the system, isolated pericytes and isolated astrocytes 2.0. All of the cell types come to you from the same donor, similar to isoneurospheres, as I mentioned before. I forgot to add that all of the cell types will come from the same donor. So again, you have this ability to match the cells closely for those of you who are working on HLA typing as well. And I'm very happy to say that our BBB system has been adapted again globally. And it's nice to see that big CRO companies like Visical have literally adapted our BBB model very efficiently with regards to the data that they generated of the model and the services they provide. And I'm just showing you some snapshot of the data that is available on the website. And just really, really beautiful to see that the data that they developed on our ISO BBB system, obviously in vitro, corresponds really closely to the data that they actually developed on the human in vivo BBB models. So again, you have a number of different compounds compared with the R square being 0.077. So again, highly correlated in vivo versus in vitro human data. And you can see, again, some information about some positive and negative controls with regards to how the compounds are able to penetrate BB system and so on. So again, you can monitor how properly the cells are connected. And again, you can screen your compounds in no time. Again, BBB system is widely adapted on transwells, but we also are working with a number of different platform providers, and we also have a number of different high throughput screening or like pretty much microfluidic systems in-house. And we do work on generating more and more information on like Mimetas platforms, Emulate platforms, Netri, and so on and so on. So again, always ask us about the platform of your choice. And this is just a snapshot of BMX on microfluidic system Mimetas platform. And again, let us know if this is something that interests you, because our team is spending a lot of resources, a lot of time on optimizing our cells on different platforms. So you don't have to do that. You just have to ask us, and that's it. And this is just a snapshot again on ICBBB in 3D spheroids, because some customers and collaborators are actually interested in having the BBB system in 3D for their specific assays. And again, this is becoming more and more easy and more high throughput screening friendly. And we are very happy to support that system. So again, let us know and we will share information with you as per your needs. So now switching the gears to cardiosphere. So hopefully those of you who are working in neurospheres or like in neuro research area will stay with us just for fun, because again, with cardiospheres, I will also show you that reproducibility is key to our to what we pretty much pride ourselves in. So please bear with me. And you will see some beautiful data on how to efficiently build cardiospheres, so like cardiac microtissues in 3D. And you can see that these cardiac tissues in 3D are, as in the case of isoneurospheres, actually do have improved maturity with regards to how the cells are responding to compounds as well. Again, for those of you who are not familiar with our isocardiomocytes portfolio, I want to tell you that the cells are incredibly established by pretty much everyone in the field. So you can pretty much come to us and rest assured that the BMS will provide you with a number of different support documents. And again, we have thousands of publications on cardiomocytes alone, to be honest. So the cells have been adapted. The cells have been commercially available since 2010. Starting with cardiomocytes, and cardiomocyte square just provides you with like a faster solution for having the cells ready upon thawing 4 days post-plating, and pretty much start using them for your compound screening. And I'm not sure if any of you are familiar with SIPA initiative from HESI, but this is a massive initiative that focuses on establishing cardiac models on MEA platforms for pretty much making sure that you're not missing out on compounds which are targeting hERG channels. So again, it's a massive initiative, and I'm very happy to say that our cells have been widely adapted and verified for their functionality and maturity and sensitivity to compounds globally by a number of different companies, organizations worldwide. And you will be able to see from subsequent slides that we do have a number of, again, options for building those isocardiac spheres. And you will see that our isocardiomocyte square or isocardiomocytes can be easily mixed with our isoendothelial cells and our new upcoming isocardiac fibroblasts to build this isogenic model of the cells. And you can also substitute isocardiac fibroblasts with primary fibroblasts if you wish, because of the historical reasons. But this is our new addition to the family, isocardiac fibroblasts. And we are in the process of releasing new application protocol for, again, efficiently generating cardiospheres from all those cell types which are coming from us. But again, we have plenty of data, plenty of applications on mixing cardiomocyte square, mixing isoendothelial cells and cardiac fibroblasts to efficiently build screening models. And again, I keep mentioning that reproducibility is key to us. It really is. For each cell type, neurons, hepatocytes, cardiomocytes, we do pride ourselves with regards to reproducibility of each batch of the cells, which means that whenever you order cells from BMS, from us, and BMS from your distributor, you will be able to just pretty much thaw the cells and be rest assured that the quality post-thaw will be highly monitored and batch to batch reproducible. So this is just a very quick snapshot of such data we can easily provide for other cell types as well. In this case, we are focusing on isocardiomocyte square from 10 different lots, looking at functional assessment, like a mean bit rate across different lots, different production lots, which require preserved at different time points, again, independently. So you can see how beautiful these cells are. And again, the number of applications, number of success stories we have is unparalleled. And the cells have been always praised for the beautiful functional quality and robustness across the lots and experiments as well. So with our isocardiomocytes, our isocardiomocyte square, we are obviously very well known for the SIPA because of the initiative from HESI. But again, we have number of globally established publications, collaborations, adaptation protocols across the slides, where the cells are not only in 2D, but again in 3D for screening, and microfluidic systems, and pretty much cardiac microtissues and so on. So again, we do accommodate pretty much any single type of research you want to do on these cells. And we have a lot of established protocols that we are freely sharing with you. So again, please ask BMS and they will be able to provide you with all this information, because they have a beautiful technical support as well. You will also see a really nice example of the publication published by AstraZeneca, where they, I will show you that in the next slide, where they actually just quickly build isocardiac spheres, cardiospheres from our cells and additional cell types, and use them for screening very efficiently. And they show that if you compare the cardiac microtissues versus 2D monolayer, the sensitivity can still be in their settings, because they're using also the primary endothelial cells and the primary cardiac fibroblasts with our cardiomocytes. You can see that sensitivity is kind of like higher for cardiac microtissues, but monolayer cardiomocytes are kind of like quite closely following the trend. But specificity is very, very different as well. So again, you can see differences in sensitivity and differences in specificity between those co-culture models and monoculture of cardiomocytes. Again, beautiful paper showing you a number of different multi-parameters readouts on these spheroids and highly high throughput screening, high content analysis fashion. So again, I encourage you to look at this data. There's abundance of information points. And again, it's a beautiful example very early on, 2018, showing that the cells are, our cardiomocytes are amenable to 3D research. These days, we are making things much easier for cardiomocytes researchers, because we have a number of our own application protocols that you can just follow in very simple steps. Again, thaw the cells together, mix them like isocardiomocyte square, isocardiac fibroblasts, and isoendothelial cells, all from the same donor. Mix them together at one day, and again, pretty much 2 weeks later, you are able to use these cells for basic and functional assessment after adding your compounds again. So it's a very simple readout as well with regards to the mixture of the cells. It's a very cost-efficient system as well, as in the case of neurospheres as well. And again, assay is super simple. You just pretty much add dye and you run the assay, and it's super easy. And again, here, for consistency across different spheres, we are focusing on calcium oscillation patterns as well. And again, neurospheres, we have to look at the core of the cells. And again, it's really, really easy to see that you can keep the cells up to 10,000 of the cells per spheroid, which is a really nice clusters of cells for those of you who actually want to use these cells for like bioprinting or more advanced culture systems later on. You can build spheroids up to 10,000 cells and there will not be necrotic core. And this, again, the data is very consistent. We usually focus on 5,000 total cells per spheroid. We can go lower again as per conversations as well. And you can see that the data is consistent and the data is high throughput screening friendly, as setup is high throughput screening friendly manner as well. And ice-like radiospheres show positive inotropic responses. So again, you can see here calcium oscillation patterns. And yellow, you can see the control condition where the cells are treated with vehicle. And in red, you can see here on the left part, the cells treated with isoproterenol. So again, as some of you may know it already, with adding this compound, the cells are going to beat much faster. And this is one of the actually quality control criteria that we use on cardiomyocyte square at cardiomyocyte. So each batch of our cardiomyocytes, before it's released to customers and collaborators globally, will be verified for functional activity on, for example, MEA. But this is pretty much equivalent data. So you can see beautiful difference before and after compound treatment. The same goes for dobutamine. And you can see that, again, ice-like radiospheres have a positive ionotropic response when treated with ionotropes. And again, this is an indication of like improved maturity. So we have thousands of data points for that. Please let us know. I had to keep it to the minimum for this presentation. But again, we really do have plenty of data, our own and external as well. So you can see how beautiful those micro tissues are. And again, it's very easy to stain them in 3D as well and build the platform based on this as well. So again, let us know. I will now focus very quickly on hepatocytes hepatocytes spheroids. And again, either in monoculture or ecoculture with macrophages, again, depending on the research that you are investigating. And again, consistency is everything to us. And this is like the simplest slide possible, just to show you that across different 12 lots, you can see randomly selected lots pretty much for the study. You can see that purity, according to our quality control parameters and post-viability, is really, really consistent. And again, the cells are beautiful in 2D. I love them. But also we have beautiful 3D application protocols that has been widely adopted for quite some time now. And you can build those spheroids and modulate the diameter very easily. And again, it's just beautiful to see how maturity and how specific functional assessment is just simply going up because the cells are in 3D, even in monoculture. So again, beautiful, beautiful setup. We have a number of different, obviously, case studies where platform providers or kit providers like Promega are focusing on using our cells to prove the point that the platform is working, the kit is working. So here you can see such example by Promega, where our iCell hepatospheres were used for showcasing the fact that the kit from Promega on glucose uptake works beautifully. And again, here the cells were treated with increasing concentrations of insulin, and obviously insulin is promoting glucose uptake and so on, is modulating glucose uptake, and you can see this beautiful correlation shown here. So again, beautiful setup, high throughput screening friendly, cost efficient, incredibly friendly. And again, it's very easy for us to provide you with information on how to kickstart your research. Also, there's plenty and plenty of data publications, internal, external publications, showing how you can use these cells for in 3D for hepatotoxicity assessment. And I will just leave you with this, with the link or information on molecular devices application and publication on, again, having these cells in 3D following our protocol. And again, just pretty much looking at number of different parameters in a very simple, high throughput screening friendly manner. You can, again, in a very cost efficient way, screen your compounds in thousands and thousands. And again, we have plenty of information on that as well, that I wish I could show you for disease modeling as well, focusing on NFL and FLD as well. So I will leave you with some data on hepatocytes 2.0, co-cultured with macrophages 2.0, because macrophages in general, mixed with hepatocytes, become those resident Kupfer cells. So again, providing this immune aspect of protecting their liver. We are recently have released macrophages 2.0, which are great on their own. This is actually the only iPS direct macrophage product on the market, and the cells are absolutely incredible with regards to their functionality. How they compare to primary macrophages is unparalleled. They have beautiful superior functionality, and they come to you in naive state. So for those of you who are working oncology, you can prime these cells to be either pro or anti-inflammatory. And again, these cells come to you at naive state, you thaw them, and literally from thaw, you can start using them for assays. And it's a really, really simple adaptation to a number of different assays you may choose these cells for. But for this, so again, contact BMS if you have interest in these cells. And again, the cells are taking our oncology market by storm. Whether you are looking at antibody detection, whether you're looking at, again, stimulating the cells with some cytokines and so on, and again, looking how the cells respond. But for now, I wanted to show you very quickly that macrophages can be easily co-cultured with hepatocytes. Again, simple protocol. And you can see beautiful spheroids comprising of both cell types, again, in a reproducible manner. And you can start using the cells 2 weeks post setup. And in a couple of weeks, we're actually going to have a number of new data points showing you compound treatment before and after the addition of macrophages as well. So again, please contact BMS if this is something that interests you. And for now, I think I'm a little bit over with time, so please kindly don't mind. But I would like to thank you for your attention and again, encourage you to contact BMS with your questions to see how easy it is to adapt our commercial available human iPS-derived cells in your research in a cost-effective and high-throughput screening manner in no time to advance your research. And again, thank you so much for your attention, and I'm very happy to take any questions on board if you have any.